Hi, I'm Ganesh Anand. I'm an associate professor in the Department of Biological Sciences at the National University of Singapore and US. I'm going to be talking to you today about infectious viral diseases and in particular COVID-19, which is a serious global pandemic now. And I'm going to be sharing with you some perspective of what viruses are and how can we observe them, how can we study them, and how can we mitigate some of the effects of these severe viral diseases. So what are viruses? And the first question would be, what do they look like? So for a long time, because they're so small and not visible to the naked eye, viruses were not known directly, but they were known as contagium vivum fluidum. So it was recognized that something in the infected patient's fluids was still contained something that was highly contagious. And so that's what was known about viruses. But in the early 1900s, it was found that these viruses were actually physical particles and that they contained proteins. So they're proteins, and what do they look like? So there's broadly three types of virus shapes that you see and variations. One of the types of virus particles that you see is long rod-like particles. And so that's the helical particles that are assembled as a helix. So each of these green droplet-like structures is a particular protein that encapsulates a genetic material, which is this wiry black strand inside the virus particle. Another type of structure that's seen is an icosahedron. And an icosahedral structure is one that looks very close to spherical, but it's formed by assembling different triangular shapes together. So if you try to put different triangles to form a sphere, you would approximate a sphere by making an icosahedral structure like we see here. So you have individual triangles that are assembled together to form a spherical structure with the nucleic acid genetic material inside the particle. And I'll come to what the genetic material is in a minute. And then there's also complex structures that you see. This is a class of viruses that infect bacteria called bacteriophages, and they have a complex structure that's shown here. So because COVID-19 is an icosahedral virus, I'm going to focus more on the icosahedral virus structures and that particular assembly. So in a nutshell, viruses come in all kinds of shapes and sizes. So you have the chickenpox and smallpox viruses, and you have herpes virus, and they all have different arrangements of proteins that are forming these particles and that encapsulating the genetic material. Now, viruses are pretty inert when they are outside the host cell, but when they enter a host and infect it, their genetic material is what allows them to make multiple copies of themselves. So all living beings contain genetic materials that allows them to make copies of themselves. So viruses appear to be non-living outside the host, but once they enter a host, because of their genetic material, they are able to make copies of themselves, and so they appear living. And so this genetic material is composed of what's known as nucleic acid. So there's two types of nucleic acids in nature. One is RNA or ribose nucleic acid, and the other is DNA or deoxyribose nucleic acid. So based on whether a virus particle encapsulates an RNA nucleic acid or a DNA nucleic acid, it is classified either as an RNA virus or a DNA virus. So when we think about viruses, I've already told you they're too small to be able to be visible to the naked eye, but how small is small? So this is a size chart which shows from 1 millimeter and smaller what are the different particles and where viruses fit into this size chart. So on one end is 1 millimeter, so a frog's egg is visible to the naked eye. And so smaller than that is a pollen grain and human egg, and even smaller than that is a typical plant cell or an animal cell. And so all of these sizes, in order to be able to view them and the detailing, you would need a light microscope. And so the bacteria are smaller than plant and human cells. And these are subcellular organelles that are found inside the cell. And viruses are much smaller than this even. And so you see a range of virus sizes. The smallpox virus is one of the bigger viruses. This is the influenza causing virus, and this is the polio virus. And these are much smaller than animal or plant cells. They're just a few nanometers in diameter, these icosahedrals. So they're bigger than proteins, but then they are much smaller than a bacterial cell. And in order to be able to view these virus particles, you need a special kind of microscope known as an electron microscope. So if you took an electron microscope and looked at an icosahedral virus, what would you see is what we're going to look at in the next couple of slides. So if you think about a virus particle, you have all of these triangles. Each of these is the protein that's forming the shell, and this was the description of the geometry was done by two scientists, Casper and Klug, and they showed and described the specific arrangement, how these vertices are formed. These are these bulges when you assemble each of these triangles together. So there's different vertices. If there's 5 points that are converging, that's the 5 vertex. Then there's 3, which gives you the 3 vertex. And where 2 triangles meet is the twofold side of the particle. So if you look at COVID-19, you'd have seen all of these images on television and in flyers. You see that it's not a smooth sphere. The icosahedron has all of these spikes which are protruding from the particle, so that gives it a very spiky appearance. So that spiky appearance is caused by a protein called as the spike protein, which is the one that's protruding out. And so this is the outermost layer. If you peel the virus, and if you just go beneath the spike protein, you have a little membrane, and that membrane is formed of fatty acids, and that's where the spikes are protruding out of. So just beneath that, you have this membrane that's formed of fatty acids, and then there's proteins that are embedded in the membrane, and you also have, if you go beneath the membrane, you have the RNA, but the RNA is packaged by a nucleocapsid protein. So all of this together gives you a compact virus particle. So SARS-CoV-2 is an RNA virus, but it also contains a membrane. And this is an animation from Fusion Medical Animation, which shows you the SARS-CoV-2, the causative agent of COVID-19. It has these spike proteins. Those are the ones that are protruding out. You can see how much bigger the infected human cell is. And once infected, the human cell becomes a factory for making many, many copies of the new infecting virus particles. So one single virus particle is able to make thousands of copies of itself by infecting cells. So we know a lot more about viruses that have been studied for a longer time. This is the dengue virus, which is also another RNA virus and a serious problem in Singapore and the tropics. It also contains an RNA genome that's packaged into another icosahedron. And this one, you can see how it infects a target cell. And the virus has specific ability to recognize something very specific called as a receptor. So it fits like a jigsaw puzzle. It fits in exactly. That causes the whole the surface to form a bulge, and that bulge causes the virus to be absorbed in a little droplet within the cell. So now this is a little droplet that contains the virus, dengue virus particle, and that's inside the cell. And once it's in this particle, it now starts to undergo these rearrangements that you see here. And these rearrangements cause the shell to dissolve, and it releases the cargo, which is the genetic material. In the case of dengue, like COVID-19, it is the RNA that's going to come out. That's here. So now once this RNA comes out, it can make thousands of copies of itself. It kills the infected cell in many instances, and those new thousands of cells can now infect other host cells. It can be spread to other patients. And so that's the thing about COVID-19. It's making many, many copies of itself. There are certain people who develop symptoms, and then there's also asymptomatic cases who make viruses, but they don't suffer any symptoms because of the virus replicating in their bodies. And so they're just as a problem in the spread of the disease. So before I sign off, I want to ask you a couple of questions based on the slides I showed you now. So the first question is, why are viruses? Viruses are considered to be both living and non-living, at the edge between the two. So why is it that they are considered both living as well as non-living? And the second question is, based on what you just learned, how does washing with soap and water help in blocking the spread of infections such as those causing COVID-19, the SARS-CoV-2? And a hint in that respect is think about what I told you about the membrane and the effect of soap there. So that's a clue as to why washing is particularly critical in preventing the spread of COVID-19. Thank you for your attention.